all of the secrets that have been hidden from our eyes for millions of years are now sort of exposed. We can use these kinds of techniques to really start to flesh out what these animals might have looked like. And so this is an animated 3D model that we did in Maya, where we reconstructed the blood vessels, the nerves, the jaw muscles, the brain. You can even see we've got the eye muscles. And if you look close, you can actually see the eye is actually turning around in there. Key part of this is we are trying to actually see how all these different soft tissues, in a sense, fit into the head.
To take that rock and turn it into that animated entity.
Scanning really sort of changed everything.
Dr. Lawrence Larry Whitmer is one of the people who helped change how we study dinosaurs as living animals, not just skeletons. Dr. Whitmer is a professor of anatomy at Ohio University's Heritage College of Osteopathic Medicine, an Ohio Presidential Research Scholar, and the Chang Professor of Paleontology. He also served as president of the International Society of Vertebrate Morphology. Before coming to Ohio University in 1995, he trained at Cornell, the University of Kansas, and the Johns Hopkins University School of Medicine. He's best known for fleshing out dinosaurs, combining classical anatomy like dissection, histology, and osteology with high-tech CT scanning and 3D modeling to reveal what dinosaurs were like in life. Much of his work focuses on the head, the brain and sense organs, feeding systems, and display structures, but he's also contributed to major studies of the neck and locomotion. He's been widely funded, published in journals like Science and Nature, mentored dozens of graduate students, and you've probably seen his work featured in National Geographic and major documentaries from outlets like the BBC, Discovery, and NHK. Before we jump into the science, I want to tell you why today's conversation is extra exciting for me. I recently got to meet up with Larry in person and spend time in the CT world with him, scanning a skull that a lot of you will recognize immediately. It's a Tyrannosaur skull that's often referred to as Nanotyrannus, and I know that name sparks debate, which is exactly why this is so interesting. A good CT dataset lets us look past surface details and ask better questions about internal anatomy, sutures, sinus spaces, tooth roots, neurovascular canals, and all the stuff you cannot see when you're just staring at bone. We ran the skull through a state-of-the-art Philips dual energy medical CT scanner, and what struck me was how fast you can capture a clean data set, then immediately start exploring the skull in slices. You're not guessing where a feature is, you're verifying it. And it was not just that skull. We also scanned other fossils that raised their own big questions. The kind of material where a CT scan can reveal hidden structure without touching the specimen itself. Watching Larry work is like watching someone translate bone into biology in real time. That's what today's about. How CT scanning helps us see the invisible, and how you do it in a way that is careful, testable, and grounded in anatomy.
And I just can't get my face out of its nose because it's just really complicated. And there's lots of stuff going on there. There clearly was really important, something to devote half their skull to. Big sort of question that I get all the time is what route does a paleontologist take? A vertebrate paleontologist or a dinosaur paleontologist? Is it a geology route or a biology route? And the reality is, don't tell anybody.
Today, we have an extraordinary guest. Dr. Lawrence Whitmer, you can call him Larry, is the legend of CT. Now, I first met Larry in 1998 in Berlin. He and Dino trained Dr. Scott Sampson were working on Berlin's Archaeopteryx using some cool lighting techniques. And I was there checking out Brachiosaurus. I rounded the corner and we ended up spending a couple nights adult beverages in various restaurants and having a grandiose time in Berlin. And we recently hooked up again because he was so kind as to bring some specimens to his CT scanner project. And just an utter delight, wonderful human being, Larry, inspirational for generations. Your lab is world renowned for producing top shelf anatomists as well as scientists in the technological realm. So, Larry, can you tell us a bit about yourself, how you got into this whole world, and anything else?
Sure. Well, thanks to you all for showing up today. And I see lots of familiar names and faces here. So for some of you folks, we've been connected on social media, and it's nice to actually connect here, sort of in person. Brian, you'd like me to go through sort of how I got here?
Yeah, so you've been, I mean, you're near, you've been running a lab for quite a while, but at one point you were Larry the student and lover of paleo. I would love to hear, the origin story.
Yeah. Well, I guess I was Larry the little kid who was just loved dinosaurs too, like maybe many of us. Some folks discover them later in life, but I was one of those people, like Scott Sampson, like you mentioned, that fell in love with dinosaurs as a little kid. There wasn't much you could do at back then. There was no internet. They're basically, libraries, these big buildings that have books in them. You may have heard of them, and museums. But there weren't as many museums, and there was really only a handful of museums that had dinosaurs. I grew up in upstate New York, and so my parents, which were very supportive, took me to the Royal Ontario Museum in Toronto, as well as the American Museum of Natural History in New York City. So those were two outstanding museums that had fossils and dinosaurs. But I didn't touch an actual dinosaur bone until I got into graduate school. So as a kid, I was interested in all this stuff, and I liked science, but I liked plenty of other things too. I did sports, and I always have done music in one way or another. I was in bands in college and stuff like that, and I still play. There's actually like over a dozen guitars in the room right here where I am right now. Probably shouldn't admit that, but scientists have other parts to their life as well, so that's worth knowing. You don't have to be devoted 100% to anything. In fact, it's good to have balance, that kind of balance in your life. But I was sort of a sciencey guy all the way through. I went to Cap College. I went to Cornell University in upstate New York. That's where I'm sort of from, Western New York, Rochester, New York, is the city I grew up in. And I was a biology major, and basically all of my degrees have wound up being in biology. And the big sort of question that I get all the time is, what route does a paleontologist take? A vertebrate paleontologist or a dinosaur paleontologist? Is it a geology route or a biology route? And the reality is they both still are legit. To a certain extent, it depends on where your interests lie. For me, I've always been interested in dinosaurs as living creatures. And so I've always been drawn to the biology of extinct animals, and the fact that they were found in rocks was interesting and informative because it tells us something about the environment that the animals lived in and obviously their geologic age. But I was always interested in the biology. So all of my degrees are in biology. I got a master's degree at the University of Kansas. That was again in biology. And then maybe surprising to many people is I actually got my PhD at a medical school. I went to Johns Hopkins Medical School, which sounds kind of nuts, but the reality is I have been in medical schools for over 35 years, because not only did I get my PhD in a medical school, but I've been teaching in medical schools for over 30 years right now. And I'm just going to sort of go through why that's not as crazy as it may seem. The reality is that medical schools teach anatomy. I'm an anatomist. I teach structure of the human body. For medical schools, they want researchers who are scholars, people that are writing, doing active research, doing publications. Something that's very important at universities is actually getting grants, federal grants. The reality is that the study of the human body is not really a scholarly pursuit anymore. We kind of know how humans are put together and have for, 200-300 years. A 100-year-old anatomy book looks pretty much like the ones today. So for medical schools to have people that have a passion for anatomy and can teach it and want to teach it, but are scholars that are publishing and doing research and getting grants, you need to get anatomists who are studying the anatomy of non-human animals. So lots and lots of medical schools have anatomists that are paleontologists or anthropologists or biomechanists, people that study the biomechanics, feeding and locomotion. And so basically all of the anatomists at Johns Hopkins when I was there were paleontologists and anthropologists, actually where Dr. Curtice went as well. A lot of them were also medical anatomists. That's also true for the Harvard MIT program, Brown, University of Chicago, Paul Sereno, I don't know if he still teaches anatomy, but he used to teach human medical gross anatomy. It's very common. And I can tell you that when I was sort of at the stage where most of you guys are, I never thought I was going to be in a medical school. But the reality is, this is my lab, which doesn't look much like a medical school. Looks more like a museum. And, there's T-Rex sitting right there, and there's all kinds of cool skulls in there. So to a certain extent, I'm kind of living the dream I had as a little kid, that I wanted to grow up and study dinosaurs. The path was a little different than what I thought. I thought I was going to be sort of, one of these guys working at a museum, every summer going off and traipsing through the Badlands and looking for that, glint of bone that leads to the next big find. And the reality is I have done that. I've done lots of field work, but I'm actually not really a field paleontologist. I have done that and I've enjoyed it. I like to think I was pretty good at it. But the reality is the research questions that I ask and what my team asks aren't really paleo field questions. In other words, I study, for example, I'm really interested in the brain and what goes on in there. But if I'm interested in, say, the brain case of Troodon, or Stenonychosaurus, one of these small bird-like theropods, to sort of say, like, you know what, I'm going to go off to the field and I'm going to find the brain case of Troodon. I'm going to go there and I'm going to find turtles, because that's what you find when you go out in the field. You find more than turtles, but you're probably not going to find a Troodon brain case. And maybe you would, but boy, is that going to be hard. Whereas I can just go to a museum and I can go see what I need. I might have to travel. I might have to get grants to travel to all these places, which I do. And so in effect, I do a lot of my research, the equivalent of what for many people would be a field season, is I'm traveling off to museums and doing that kind of stuff. If anything, some of the field research that I do these days is actually looking at modern day animals. Many of the questions that we're asking about dinosaurs actually need to be answered by looking at animals that are around today, extinct animals. And so we're really interested in sort of the physiology, how animals deal with heat in particular, and how they cool their brains, how they cool their sense organs. And so we've done stuff like, gone down to Florida with thermal imaging cameras and studied like pelicans and crocodilians, and study the blood vessels and how they shuttle heat around the body. And so in a sense, that's kind of the field work that we do. Questions often arise about dinosaurs, but to answer them, we need to look at their modern day relatives. So my path here is maybe a little different than most people. But it's actually not as unusual. This whole medical school thing, I bet none of you, maybe you know that some paleontologists are at med schools. but I never thought about that. I basically got into it as a graduate student. So as a grad student, kind of the cool thing about grad school is that you often get paid to go to grad school, in the sense that you often get tuition is covered. That's fairly standard in grad school, not universal, but pretty common. And you often have some sort of a stipend, an assistantship of some kind. And very often those are teaching assistantships. So many of you in college have had TAs that have run labs or done things. Very often, if there's a museum at Kansas, there was a museum of natural history. And so I had a curatorial assistantship. So I was in ornithology, numbering bones. I actually numbered a hummingbird bones under a microscope. And that gave me money. And research assistantships, where you're working on someone's grant. I have had grad students working on my grants as research assistants. And when I was a grad student, I was also a research assistant. And so how I got into anatomy, it was a TA gig. There was an anatomy course that needed teaching assistants. And so I took it, and I discovered that it turns out there was a whole bunch of jobs, assistant professor jobs, in teaching anatomy. And the reality is that any university, any college has some kind of anatomy. So I'm in medical schools, but there's also like undergraduate, there's like nursing and anatomy and physiology courses, physical therapy, there's all kinds of courses. So everybody teaches anatomy, somebody does, human anatomy. And so it turned out to be a good gig. And all of the students that have gone through my research lab have become excellent paleontologists, excellent comparative anatomists, but they also, in a sense, learned a trade. I viewed that as something I did as a graduate student, is I learned a trade. The fact of the matter is, the job I have here, they weren't saying like, hey, we need a dinosaur paleontologist. What they said is, hey, we need somebody to teach anatomy. We want them to have research that actually is productive and all of these things, done at a high level, blah, blah, and also jives well with other scientists that were here. And so my research area was viewed as actually fitting in and being appropriate for this group. But the reality is, I got the gig because I could teach human anatomy. And that's something that, but I also had to be that good scholar. I also had to be a scholar that was publishing and was either getting grants or had a high, I was young, so how many grants am I going to have had? But I had the prospects of getting grants. And so it's really helpful to have both of those sides. Those of you who are thinking about grad school, you should expect to be trying to pick up some of these skills that make you marketable. You don't want to just think about being, you want to be a great scientist, but somebody's got to pay you at some point. And they will pay you to sort of be a great scientist, but the job often is to do something else. Often it's teaching at universities. And at museums, it's doing museum work, which is only partly research, as you might not know that museum people do other things as well.
So I've got so many questions, but when you were at Johns Hopkins, was it Weishampel, or were you there with him as a student? How did that work?
Yeah, so I was Dave Weishampel's first student. You may have heard of Dave Weishampel. He's retired now, but he was a hadrosaur worker. He was the senior editor of the Dinosauria, which has gone through 2 editions, and there's a 3rd edition coming. And so, yeah, I was his first student. He was, I've got, a sibling that's his age. So he and I were, not that different in age. I was his first student. So he was learning to be an advisor as I was learning to be a PhD student. And, we sat around and listened to Grateful Dead bootlegs and stuff like that too. So we were similar enough in age and interest that we were sharing more than science, sharing music and other sorts of interests at that time.
And from there, was it Larry Marshall, or who were you at KU with?
Yeah, Larry Martin. Yeah,
Larry Martin, sorry.
yeah, it's okay. Larry Martin was my advisor at the University of Kansas, um, and he was very supportive. He was very supportive of sort of this extant, this modern day side that I was really pursuing as well. I was fortunate that at Kansas, I literally was in a museum. So it was a museum of natural history. My office was in the paleo part, the vertebrate paleo section of the museum, but there was herpetology and collections and mammalogy, and actually one of the top um, ornithological skeletal collections on the planet, which I didn't really appreciate until I left, because they had amazing collections. So in a sense, I had a really great formative experience being in a museum, as well as at a university. And so that really gave me a view of sort of all of that side. There were exhibits, and the museum faculty were faculty and teaching classes, but they were also dealing with exhibits and working on exhibit text and designing exhibits and things like that. And so that was very interesting. And I've always had interest in being in a museum. The reality, I don't know what the actual numbers are, but there are many, many times as many university and college jobs as there are museum jobs. If you think about it, there's way more colleges and universities than there are museums. And those universities and colleges have a lot of faculty. And each museum has just a few curators. They may only have one curator of vertebrate paleontology, or maybe two or three if they're really big. And so museum jobs are there. They're just not as common. But one of my students is in a museum. One of my students, Catherine Early, is now an ornithology curator at the Science Museum of Minnesota. But actually, all of my other students, other than Catherine, are anatomy professors at medical schools. So, but they're paleontologists doing stuff like me. Casey Holliday, who you may have heard of, is one of my former students. And so, you know, it's there's sort of many different paths, and you know, I got lucky. I also recognize that I benefited from, you know, the privileges I had of birth. I was, you know, born at a time when doors were opened for me that maybe weren't as open for other people. I'd like to think I, you know, was good enough to open doors for myself, but the reality is there were doors that were open for me that were not open for others at that time. And I'm glad that the culture has worked on trying to open doors for everyone. I've tried to do my part for opening those doors, but I recognize that I had an easier path than maybe many other people of my generation that looked different than I do.
Well, the fact that your lab keeps turning out anatomists, that's kudos to the, you know, your college is probably happy with you that you're still, the apples are falling close to the tree and you're sharing your knowledge.
You bet. Yep. No, they like that.
And with regards to what you're working on, so how did you get into computed tomography, aka CT, if that's even what it stands for?
Yeah. CT scanning. Sometimes people say CAT scanning. That's an older term, but you'll still hear that. I didn't start out with that. So CT scanning, computed tomography, it's an X-ray modality. I've got some slides I can show you about that in a second. I mean, it's been around for a while. It really started in the 60s. What a lot of people don't know, I don't know if many of you are young, so you might not have heard of a famous rock and roll band called the Beatles. John, Paul, George, and Ringo. But I like to say that the Beatles actually invented CT scanning, which isn't entirely true, but it's kind of true. And the story with the connection with the Beatles and CT scanning is they had a company called the parent company of Apple, which was the Beatles record company, was called EMI, Electronic Musical Instruments. And it turns out that wound up being a pretty big company that was making lots and lots of money because of the beetles. It turns out they had a medical arm as well, that company. And there is a guy there named Hounsfield, a scientist who actually won the Nobel Prize in Medicine for generating the first CT scanners. And he did that based on money that ultimately came from the beetle. So it's a little bit more complicated than that, but I like the idea that the beetles invented CT scanning. It's a connection I have to the beetle.
They
So
more
the
than
CT
just held
scanning
your they more than just held your hand.
That's right. That's right. And so, yeah, CT scanning. And this is actually an interesting part of a career as well. And so, although CT scanning was obviously around, it was around when I was a little kid, but it was rare in hospitals and really didn't exist as a research tool outside of medicine. And it really wasn't even a research tool in medicine until later. And so I was pretty much a professor, a young assistant professor, when people started to do CT scanning. So I was one of the first people, not the first, but I was one of the first people that started to do a lot of CT scanning and 3D visualization. And so that was an interesting thing for me to do as, in a sense, a fairly established scientist, which I had to actually respond and learn new things. I'd learned things as a professor that helped shape my career. All of the learning I did wasn't done as a student. The reality is that, and it's one of the things I love about being a professional scientist, is yes, I train students, but I regard myself as a student. I'm still learning all the time. The world is changing. I'm still studying right alongside with my students, and it's not uncommon for my students to become the teacher. Certainly, there's newer techniques that they're hipper to than I am, and that's how actually I was too. I got sort of in on the leading edge or front edge of CT scanning, and I was working with more senior colleagues and pulling them along into the 21st century. And so CT scanning really sort of changed everything, to be honest with you. CT scanning uses X-rays, and early on when we were doing CT scanning, all we really wanted to do was look inside to see what was going on inside, to see what was going to, in a sense, to peer through the bones of the fossil, to peer through the rock that was still surrounding the fossil, to see what was there. And that's still super important. But one of the things that is key, and Dr. Curtice's background is showing that nicely, is that by CT scanning, and there's Diplodocus in there, as well as the Cleveland Tyrannosaur, Nanotyrannus, or probably a juvenile T-Rex, depends who you talk to. It's CT scanning, in a sense, brings that object, that fossil, into a 3D computer environment. And so, yes, we can peer inside thanks to the X-rays, but it also is a way of, in a sense, digitizing these fossils, bringing them into a computer environment where you can do things to them. You can digitally remove the rocks, digital preparation, but also you can do all these other things. And my team has done a lot of that. You can do biomechanical modeling of bite force, and we also model airflow, computational fluid dynamics. You can, in a sense, do crash tests and run simulations and study locomotion, all of these things by, in a sense, digitizing. And CT scanning is just one of the tools we have for bringing a physical object like a dinosaur fossils into a computer. There's also laser scanning and photogrammetry, and those are all excellent techniques. But CT scanning is special for me because it allows us to look inside. And a lot of the stuff that I really want to know about dinosaurs is inside the bones. I want to see the brain cavity. I want to see canals where blood vessels and nerves ran. I teach anatomy, but I can tell you I am a comparative anatomist. I am all about those little bony doodads, those little bumps and grooves and canals and stuff. Man, that's my money. That's what I study.
So do you have some examples that you may be able to show us of some of your work?
Sure. Actually, let me, I'm just going to run just a brief presentation that shows you, in a sense, sort of what we're trying to do. So I'm going to show some of these are slides that I often use at the beginning of talks, just to give a perspective as to sort of what we're kind of all about. So let me fire this up here, and we'll see how this runs. It also may depend on how you are viewing the Zoom. So I'm going to try and share my screen. So hopefully you're looking at my slides right now. It's a title slide?
Yes.
Cool. Okay. So here I am. I'll just, thanks, Brian, Dr. Curtice, and Fossil Crates for inviting me. I'm happy to be here. There is my contact information for those who would like to get me. You can just search Whitmer Lab, and there's lots of things that will come up. Let's see, I'm trying to advance to my next slide here. Being a little problematic. Give me a second here. Worked before. There we go. So this slide is basically showing, where we're starting and where we're trying to go. So we're actually trying to go from sort of bones, the skeletons that you find in the ground, to a fully functioning biological entity. And really, we're sort of interested in kind of knowing what kind of science is involved and really reconstructing the biological attributes of these extinct animals. So where do we start? We start with bones in the ground, which we carefully remove and which we may mount in very lifelike poses in a museum. But we still kind of have a pile of bones, although maybe arranged in a more lifelike manner. But the reality is, a lot of fossils, like this beautiful T-Rex skull right here, hide a lot of their secrets away around they hide their secrets within the rock in which they're entombed. And so I call all of these dinosaurs that we see here on the left digital dinosaurs or analog dinosaurs, because they exist in real physical space. These are the ones that you can hold in your hands and turn them around, and you can, smell them and touch them and taste them if you want. But there's a lot that you can't do with them. And so my team has made the jump into the digital realm. You can take a skull like this T-Rex here at the bottom left and run it through a CT scanner, like at your local hospital, or in this case, one of NASA's rocket engine CT scanners. And that step actually takes that analog dinosaur and it transfers it into the digital realm. And once it's in a computer, you can spin it around and look at it from all sides, which is pretty handy for big guys like this that are almost impossible to really work with. But the key step is that we can look inside. All of the secrets that have been hidden from our eyes for millions of years, really, are now sort of exposed, and sort of that dusty old pile of bones starts to almost come alive, brimming, even dripping with biology. So we spend a lot of time looking at dinosaurs, but one of the things that we found is that if we're interested in dinosaurs, we can't just look at dinosaurs. We also have to look at the modern day relatives of dinosaurs, particularly if we're interested in looking at soft tissues or other unpreserved attributes of these extinct animals, like their physiology and behavior. And as you may be familiar, dinosaurs, are not actually extinct. We've still got birds. We still have thousands of species of dinosaurs around us today, including things like ostriches, but also hummingbirds and sparrows and things like that. We also have crocodilians, which are the next most closely related group, and lizards and turtles and mammals like us are there too. We're more distant related, but we also are relevant because many mammals are in the size range or are ecological analogs for dinosaurs. And so the key thing is that if you're interested in dinosaurs, you can't just look at dinosaurs. This is sort of a slogan: the present is the key to the past. That's something that has governed geology and paleontology for over a century. And what it means is that if you're interested in dinosaurs, you kind of have to have one foot in the past, but you kind of have to have the other foot firmly planted in the present, where we can start to, in a sense, flesh out these animals. So the techniques we use actually involve a lot of modern day animals. We use a lot of these time-honored techniques like dissection, or we inject the blood vessels, or even do sectioning like you see over here on the right, either with histology or with bandsaws. One thing I'll quickly point out here is that all of the animals that we get into our research lab died for some other reason. We don't actually kill anything. Animals are dying all over the planet all the time. You just have to know how to get them, and I do. And so I've got a walk-in freezer loaded with carcasses that are donated to us, or working with wildlife rehab facilities that love to work with us, because we can often provide an afterlife for animals that they are unable to rehabilitate and send back into the wild. So a lot of the questions we ask about dinosaurs need to be answered by animals that are around today, that still have soft tissues that we can dissect, physiology we can measure, behaviors we can observe. But we also use techniques of CT scanning. Again, this is an X-ray technology. You can see at the top left here, that's a regular old hospital scanner, where we actually CT scanned the holotype skull of Tyrannosaurus Rex, which is what I'm holding on there. And there was a question that we had asked about Emily Caggiano, and actually there's a picture of her, two pictures of her right here. That's Emily at the lower left here, was an undergrad who was working in our lab. And there's Emily at the top right with our micro CT scanner. We have a micro CT scanner. I'm director of our micro CT scanning facility. And there's a barn owl that we CT scanned that we, in a sense, digitally plucked. We, in a sense, removed all of the feathers so that we can actually see. Barn owls are interesting because they have asymmetrical ears that allow them to really pinpoint the prey in life. And so these imaging modalities allow us to, in a sense, look inside. You can sort of see the skeleton of this albatross down here that Emily is scanning over here. Here's this holotype lower jaw dentary of T-Rex, and you can actually see the replacement teeth in here. You can also see how they implanted over a century ago supporting rods to help actually mount this thing. There's an iron rod going through there so they could mount it. So I'm just going to show you really quickly how we do some of our studies to do what we sort of call fleshing out these animals. And so here's Diplodocus, the sauropod dinosaur, Majungasaurus, the theropod. Let's focus here on Diplodocus for a second, just because it's one that we've done a lot with and one that we've focused on recently. And so where we start, of course, is with the actual fossil. This is the skull of Diplodocus from the Carnegie Museum that we got on loan. And it's the fossil, of course, this one looks pretty nice. And this is a nice three-dimensional skull, but it's actually pretty beat up, as it turns out, which we'll forgive it because it spent 150 million years living under a mountain and shows some signs of wear. So what we can do is our first step that we often do is to take this skull and we run it through a CT scanner. And that step right there actually transfers that analog dinosaur into the digital realm. And so here's Diplodocus, and it turns out that the scans that I'm looking at right here are actually the scans that Dr. Curtice and I collected just 3 weeks ago. And so I have scanned this skull of Diplodocus 3 times because technology changes, technology gets better. And so I scanned the skull in the 90s. I scanned it 20 years ago in 2003, and then we scanned it just 3 weeks ago on this CT scanner that you can see right here. And we got some beautiful results. But one thing we can also see is that it looks pretty nice, but you can also see all kinds of cracks and it's distorted and missing parts. And so what we can then do is by pulling it into this 3D computer environment, we can start to, in a sense, try to reverse the ravaging effects of time. if we're trying to flesh out dinosaur, we don't really want to do it on that beat up old skull. We want to use the skull, or a skull that's more like what the actual animal had in life. And so what you see here is a restored skull of Diplodocus based on multiple specimens, CT scans, original fossils, the casts, the literature. And we use programs like Maya and Blender to, in a sense, give us the actual skull that the animal had in life. And so people can quibble with any of the decisions that we've made here as how we restored it. But thankfully, given the variation that occurs in natural populations in life, I'm pretty sure there was an animal running around 145, 150 million years ago that looked just like this. And so, this started out with CM11161, but what we've got here really is no longer that specimen. This is just a model of Diplodocus or diplodocene sauropods. So how do we flesh these guys out? So if we go back to a fossil here, this is a different skull than we just looked at, but this is another one that we CT scan. And so what I'll do is I'll slice this skull right through the middle here, and that reveals this cavity in the bone. It turns out that cavities in the bone wind up being really important to us. And so that cavity is actually the brain cavity. Then we can use specialized kinds of software to, in a sense, fill up that cavity, giving us something called the brain endocast. And if you still got a little picture of me there, I'm actually holding a 3D print of the model of this brain endocast right here. And so basically, it's our studies of the modern day relatives that allow us to, in a sense, decide what parts of this cavity that we have in the skull actually pertain to the brain, which is in sort of light blue, versus other things. So cranial nerves are depicted in yellow here. This big red thing down here is actually the carotid artery that's supplying blood to the brain. This sort of pink or rose structure here is the inner ear that has the organ of balance as well as the organ of hearing. And dark blue are blood vessels, veins that are running through here. And so we can use these kinds of techniques to really start to flesh out what these animals might have looked like. And so this is an animated 3D model that we did in Maya, where we reconstructed the blood vessels, the nerves, the jaw muscles, the brain. You can even see we've got the eye muscles. And if you look close, you can actually see the eye is actually turning around in there. And so a key part of this is we are trying to actually see how all these different soft tissues, in a sense, fit into the head. And so we'll set that presentation aside for now, although we can go back to it if you want to look at any of those in more detail. But you can sort of see sort of the vibe of what we're going at here. We're really trying to, we sort of talk about, fleshing out the past. It's a little bit of a pun. We're trying to really see what in sort of biologically these things are like. But also what we were trying to do is kind of almost literally flesh them out. We're trying to put the soft tissues back on these things that used to be there.
That is mind-blowing, to take that rock and turn it into that animated entity. And then the next generation, they can lay skin over the top of it and make running around on your screen in prehistoric planet.
That's exactly right.
So there are a myriad of questions that have teed up in the chat.
Yeah, I see this.
So I'm going to just start from the top here. This is a question about dissecting with Emily on the evidence of large display scales and features present on the sides of the maxilla of some adult T-Rex specimens. How soon and where is that getting published?
Yeah, that's a wonderful question, and thanks for bringing up Emily Caggiano, who's this undergrad that worked in our lab. She worked in my lab for all 4 years of her undergrad, but she was actually a cancer scientist. She's actually getting her PhD down in Texas and one of the top cancer biology labs down there. But she basically said we were a side hustle. She did a ton of dissections in our lab, and so we actually started this video series called Dissecting with Emily. But then we got the holotype of T. freaking Rex on loan, the skull. And so that was in my lab for like 2 years. And so Emily and I did a bunch of stuff with that, because why wouldn't you? And she did a very popular video, and it's actually, she did a great job with that video, talking about a lot of the features that relate to the soft tissues covering the skin or covering the skull. And there were these different zones that we talked about, including some large scales, very interesting scales that, or some depressions in the skull that probably suggest that there were some interesting scales that may have been there for display. And yeah, we've got some work going on that. So when will that be published? It's a good question, because we've got parts of a manuscript and figures. You know, we got a lot of manuscripts with figures, and so it's we got lots of things that we're trying to kick out the door. I just got another manuscript from Casey Holliday that came across through email today that I got to work on too. So, but that's actually really interesting work that we're trying to get out. I know a lot of people are really interested in that because of how it affects their appearance. And we've done lots of work that relates to how dinosaurs might appear, which has a big implication for dinosaur art, which I recognize. But in many cases, we're kind of more interested. We're not as interested in the art side as maybe a lot of people would like us to be, because we're more interested in some of these other scientific questions, like, you know, what these things might mean for physiology and behavior. And those do interlap. There's a Venn diagram that overlaps with dinosaur art, and I get that. And we'll get to some of these issues. I know folks just are nuts about things like lips and cheeks and stuff like that, and we've got thoughts on that, but we're actually tackling some other questions as well. But that's a great question, and thanks for allowing me to give a shout out to Emily.
There's a part 2 of it. How would you go about looking for evidence for certain types of soft tissue, like dewlaps or inflatable nasal sacs?
Yeah, and we've spent a bunch of time on that. We had a grant called the Dino Nose Project, speaking about the inflatable sacs, that was looking at not necessarily inflatable air sacs kind of things, but that was on the table. What we were looking at are some of these dinosaurs that have a really huge narial region, meaning the part of the nasal cavity that's just on the outside, associated with the nostril. And for us, it's this part right out here, the part that you would stick your finger in. I'm not going to do it, but you can do it at a private time. That's your narial region. And it turns out in us, it's the smallest part of the nasal cavity. But in some animals, including some dinosaurs, it's the largest part of the nasal cavity. And something like Edmontosaurus, which is lurking back there, the narial region is half the skull. If you take away the frill of Triceratops, the narial region is enormous. You can stick your entire head through it. I've done it. Don't tell anybody. And it's almost half the skull. Diplodocus, if you look over Dr. Curtice's shoulder here, you can see that big opening on the sort of opening in between the eyes on top of the skull there. But the actual narial region, the nostril region, extends far forward. And so once again, the narial region is about half the skull. And so we actually got a grant to study animals with big noses. And so we actually studied a lot of modern day animals. We actually studied moose, hooded seals, which are seals that are actually have an inflatable air sac. Saiga antelope are animals that have a proboscis. We studied tapirs, which are sort of pig-like relatives of horses that actually have a short trunk. We actually published articles on all of those. They're mammal articles that actually don't even mention the word dinosaur. You don't need a dinosaur to make a tapir really interesting. And so we discussed, discovered discussed the anatomy of the nose of moose, and saiga and tapirs and all of these different things. And so once again, what that highlights is we wanted to understand. So for example, some people have thought things like Diplodocus had a trunk because that nasal thing seems retracted. The nasal opening seems retracted, like you see in a tapir or an elephant. And it turns out we actually figured out or looked at how animals that have trunks build them. And it turns out that how they build them is different than what these dinosaurs are doing. And so there are objective reasons why we can say that dinosaurs didn't have mobile trunks, or at least not in the way that mammals do. And something else must be going on. One of the things that we found is actually that the venereal region of things like sauropods, like Diplodocus or Giraffatitan, or any of them really, as well as any of the ceratopsians or hadrosaurs, had a highly vascular nose. You may have gotten a bloody nose pretty easily at some point because your nose also has lots of blood vessels in it. It doesn't take much for it to bleed. It turns out that animals use the blood vessels that are running through the nose and other areas like the mouth, the oral region, as sites of heat exchange where they dump heat. And the reason for that is that there's evaporation that takes place in the moisture in your mouth as well as in your nose. Just like the sweat on your skin cools you down because evaporation cools things. It turns out that a lot of animals will actually use the blood vessels that are going through the oral cavity, the mouth, the nose, and also the eyes. It turns out that the eyes are another moist area where we get evaporation and evaporative cooling. And so a lot of animals actually will cool the venous blood in the eyes, the nose, or the mouth, and then run that cooled venous blood back to the brain for brain cooling. And so that was one of the things that we discovered with dinosaur noses is they didn't have trunks. They actually had air conditioners. And even things like armored dinosaurs, ankylosaurs, one of the things that we published on a few years ago, was that they have really convoluted nasal passages that almost certainly functioned as a heat exchanger, maybe as a resonating chamber too, may have actually affected their voice. We can talk about that, but there are large blood vessels associated with it, almost certainly related to cooling blood that was actually keeping the brain relatively cool. So as we're trying to understand the soft tissues, we are looking at all of those systems. We are constantly going back and forth between dinosaurs and their extant relatives, their modern day relatives. You can say what you want about dinosaurs, but unless you're just making stuff up, you got to ground it in something. You got to ground it in animals that we have around today. And so on the one hand, you're trying to use modern day animals to, in a sense, ground your inferences, ground what you think dinosaurs might be doing. But you also need to allow dinosaurs to be the crazy, unique animals that they were. And so one thing you don't want to do is reconstruct dinosaurs with a very ordinary modern look. And so what we try to do is really try to balance what we do and trying to, in a sense, also allow the uniqueness of dinosaurs to emerge. But as scientists, we still need to be able to understand how these anatomical systems work. And that helps us understand what dinosaurs might have done. We know that dinosaurs would have built their elaborate structures using the tissues, the substrates that animals have around today. They're made from flesh and bone. So we want to know about like the flesh part, the muscles and the blood vessels, the connective tissues, all of these things that go together. What do the grooves and bumps and tuberosities on the fossils, what are they telling us? That's the job that I have, that my team has, is to try to figure that out.
And how do you go about estimating things like muscle and soft tissue around the skeletons of the extinct dinosaurs?
Yeah, it can be hard. I mean, so the modern animals give us something of a guide. But if you look at, I mean, everybody looks at the frill of Triceratops or the horns, and I just can't get my face out of its nose because it's just really complicated. And there's lots of stuff going on there. There clearly was really important, something to devote half their skull to. And so, you're looking at all that stuff. You're looking at all of the little details. And for us, what we want to know is, who are the players? We know them by name. So when we think about blood vessels and nerves, we're not just saying blood vessels and nerves. We're saying the maxillary division of the trigeminal nerve. We know the name of that dude. We know what it looks like in all of the modern day relatives of dinosaurs that, in a sense, bracket the dinosaurs themselves. And so we keep track of all of those different name structures. Graduate students in my lab have done entire doctoral dissertations, devoted their graduate research to these different systems. So, for example, you mentioned, how do we figure out muscle? Well, Casey Holliday spent years looking at jaw muscles, trying to understand how jaw muscles in birds, crocodilians, lizards, turtles, attached to the skull. What were those muscles? What were their names? And how did those different muscles evolve in the different groups of modern day reptiles? And the key thing is, what were their osteological correlates? In other words, the bony signatures written into the bones by those soft tissues. For muscles, we often call them muscle scars. Excuse me. Blood vessels and nerves tend to leave grooves or canals. And so we become intimately acquainted with all of those bony signatures in the modern time plane, where we can actually do dissections as well as CT scans to know who those players are and how they, in a sense, literally write their presence into the bone. The bony signatures written in by those soft tissues. And that's my job. That's really what I do. CT scanning is one of the tools I use to do that job. That allows me to reconstruct soft tissues in dinosaurs, which helps me reconstruct their behavior and their physiology and everything from there.
Does the CT scans let you know, for instance, with theropods, the question was, which have acute sense of hearing versus smell versus just a good sense versus great?
Yeah, and that's good. It's hard for us to know. Like, some people sort of say, like, T-Rex had big olfactory bulbs. Could it smell blood from 6 miles away? And it's sort of like, well, we can't tell you that. We can tell you that the T-Rex had a better sense of smell than, or was smell was more important to T-Rex as a sense than it was in some other guys. So, for example, if you can see this thing here, let me see if I can bring it up here. This is actually the brain endocast of Troodon. So this is of a or Stenonychosaurus. So this is a very small bird-like reptile, but that's a pretty good size brain endocast right there. It's very brain-like. And there's the olfactory region right there, which is pretty good size. So the sense of smell was moderately important for Troodon. But its hearing organ, you're not going to be able to see it too well back here, but this red thing right here is going to be the inner ear. And it actually has a remarkable hearing organ right here that actually curves underneath the brain stem. In most dinosaurs, it sticks straight down, but in Troodon, it curves underneath the brain stem. The modern day animals that we see that in are, in fact, owls that have a tremendous sense of hearing. And so did Troodon and its kin have hearing as acute as owls? Hard for us to say, but one thing we can say is that Troodon almost certainly emphasized hearing as an important sense for it. Also, it has an enormous eye socket. We can't see it right here, but this whole indentation of where the brain shape is right here is indented by a very large eyeball that was in there. We can also look at the size of the optic nerve, which is the nerve that actually brings impulses from the retina back to the brain. And we can look at the size of the optic nerve as a measure of the importance of vision. Can't really always tell us about, so for example, we can look at right here. Here's the brain endocast of Parasaurolophus or Parasaurolophus, one of these duck-billed dinosaurs with the big crest. And here's the optic nerve right here, which is pretty big, suggesting that the sense of vision was actually important for Parasaurolophus or Parasaurolophus. What we don't necessarily notice is that we know that vision is important, but there's two kinds of things that relate to vision. One of which is acuity. In other words, how sharp is your vision? What kind of details can you pick out? But the other part is sensitivity. How much light do you need? And it turns out that animals that live at night need more sensitive vision. In other words, they can capture photons. So you can see in low light, but that often comes at a cost of visual acuity. And some of that relates to things like cones and rods and structure of the retina, which we don't need to get into. But it can be difficult just by looking at the size of the optic nerve to see that. I can tell you that vision is important, but we can also look at something, I don't have an example of it here, but the bony eye ring. We don't have bony eye rings, but a lot of these dinosaurs did. And the structure of that scleral ring, that bony eye ring, has been shown to actually be different in animals that live in the daytime, that are diurnal animals, versus animals that live at night, that are nocturnal animals. And so we can use all of these different clues that we can pull in. We can use the structure of the brain to tell us something. We can also use other aspects of the skull and other aspects of the biology to try to make sense of it. So, for example, we can look at the brain. I don't have the brain of an ankylosaur here, but if you look at something like Ankylosaurus or Euoplocephalus, they have a pretty good size olfactory bulb. So the part of the brain that is receiving impulses that relate to the sense of smell. But if you look at the skull, you can actually see that they have a very large, complicated olfactory cavity. In other words, the part of the nasal cavity where the air comes in carrying the little particles of smell. So there's a couple of different ways you can look at the sense of smell. You can look at the part of the brain, like in Troodon here, the olfactory bulbs, or you can look at the nasal cavity itself and see where the part of the nasal cavity where smelling takes place. And we do both of those things in our lab. We're trying to reconstruct as much of the apparatus as we can. And for looking at the nasal cavity, which we've discovered is this very multi-function structure. It's involved in smelling for sure, conducting air in for breathing. It does that as well. It conditions the air. It's involved in physiological functions like brain cooling. And so teasing all of that apart is partly why I have training as a biologist, because those are the questions that I'm trying to figure out. And the cool thing is that we're kind of getting there. We're figuring some of this stuff out. Lots of questions. And I sort of view that in some ways, part of what my job is, and has emerged to be, is trying to figure out the limits of what we can actually know. There's many things that we want to know about the past. And the question is how can we know those things? What's out of reach? What things can we not know? And it turns out that we can often push back that wall of things that we can't know. We can start to know new things. Some of that comes from new technologies. CT scanning moved that wall. All of a sudden, I can know more things. But also these comparisons that we're making, this just old-fashioned comparative anatomy, asking the right questions, making the right comparisons, allows us to answer things about these and discover things about these extinct animals that we never really thought we could before. And it's a really exciting time because we keep moving that wall. Still seems like a lot of the things we want to know are just outside that wall. And I'm looking for the next thing so I can keep pushing that wall back so that we can know more about the past. And that's sort of my job, really. CT scanning, dissecting, all of these things are tools. But as I try to, in a sense, paint a picture of the past, I'm trying to do it with a broad brush, but I'm trying to fill in details with a fine brush wherever I can. But I have to also recognize that there's some stuff that we're probably just never going to know. Some of these questions, if we really want to know them, we are going to need a dial of destiny, a time machine. Nobody's actually seen that movie but me. But it's we're going to need a time machine for some of these things. But what's cool is that we're getting there. In the absence of that time machine, we're able to do much better than we thought we were ever going to be able to do when I was the age of many of you folks out there. So I'm excited to figure out what you guys are going to learn.
So, okay, first of all, I have to be respectful of everyone's time. We're at the top of the hour. So your time in particular, Larry, we would love to keep, there's a bunch of questions queued up still, but I know you have a day. So I want to be respectful of your time, first of all. And we can always have you come back too, if you're interested. So
well, we can go if you're a little bit longer if you want.
Yeah, there's still a bunch of questions. Second of all, you are bringing scientific rigor to an otherwise speculative part of the world. And at the same time, you're helping modern biologists out because you're looking at animals that, oh yeah, we can see them, like the Seriema. So I was involved with Taylor Oswald. I saw he's on here. We were involved in the Seriema project. Turned out no one had really spent any time. People had looked at the Seriema, but there's nothing in the literature. There was no videos. There was nothing we could look at. And we've got 9 of them in the greater Phoenix area. I was shocked to see modern animals don't get the love that paleo often gets.
Yeah, no, it's entirely true, and it's one of the things that we have often sort of said. We've had some scientific problem that we're looking at. We're kind of going like, well, surely someone has looked at this, and it turns out that nobody has. You mentioned Seriemas. We've got major projects going on terror birds, and Seriemas are the modern day, the closest living relatives to terror birds, and they're kind of like terror birds in a lot of ways. I've got a video that I have up on there where I've spent time with Kevin, a Seriema named Kevin, who has just taken this little toy crocodile. I've got videos that I've posted, and he's just slamming this thing down on a rock, slamming this toy crocodile down on a rock. And that's actually one of the things they do. That's how they kill their prey. And did terror birds do that? Well, actually, maybe. We've looked at some of the biomechanics of their skulls. We published that way back in 2010. We've got projects going on their brain structures. They actually have really large brains, kind of crazy big for birds. And so these animals look like they've got all kinds of tools. But you're absolutely right. We have had to go out and try to figure out a lot of and what's kind of cool is that, and that's one of the fun things about dinosaurs that have really benefited scientists, is that dinosaurs capture the interest, not only just because they're big and terrifying and kids like them, but they capture the interest of other kinds of scientists that wouldn't call themselves paleontologists. People that are like engineers kind of go like, how can you be, 40 tons and walk around on land? Physiologists are kind of going like, how can you actually pump blood to a head that's 30 feet away from your heart? And so it just recruits all of these people that have expertise that I'm just trying to get. They already have it. And so it's been this fantastic thing to actually recruit these people in to shed light on dinosaurs, which has made it much more exciting for all of us.
There was a paper recently about cassowary casks, using them to cool themselves and shed excess heat. Could the same thoughts apply to early theropods like Dilophosaurus, Dinosaurus, Oviraptorosaurs, Corythoraptor?
Yeah. I mean, and so many of these things are multi-purpose organs. So, for example, people will look at like the crest of Dilophosaurus, or any of these animals that have crests, or the frills of Triceratops. Those are clearly display structures. We know that dinosaurs were visually oriented animals, in part because they evolved from visually oriented animals, but also so many of them just have these wacky display structures. What's interesting, though, is if you look at the frill of Triceratops or any of these horned dinosaurs, ceratopsians, they've got spikes and all of these horns and all of this stuff like that. But if you look at it, they've also got all of these grooves that are for blood vessels. And so, yes, they were display organs, but I promise you, they were also heat exchange organs, thermal organs. To a certain extent, they couldn't prevent that function. Those horns, that frill, that crest of Dilophosaurus had thermal consequences, whether the animal used it or not. In other words, it was hanging out there. The plates of Stegosaurus had thermal consequences, whether they were there for heat exchange or not. And the reality is, if there's an organ that can serve as heat exchange, an animal's going to use it, because heat, it turns out, is a big deal, whether you're a warm-blooded animal or a cold-blooded animal. So many of these organs were multi-purpose organs. They were display organs. I mean, the nasal cavity, I was ticking off all these functions of the nasal cavity. One thing I didn't mention was actually vocal resonance. In other words, many dinosaurs, hadrosaurs are famous for this, the Lambeosaurus, for taking the nasal cavity way back and forming these resonating chambers. But the reality is any of our nasal cavities are resonating chambers. Any space that's connected to the airway and the vocal apparatus will have resonant properties. You can't avoid it. I can just cover over my mouth like this and turn my hands into a resonating chamber and my voice changes. So the curly Q, crazy straw nasal cavities that we see in ankylosaurs, like Ankylosaurus, Neoplocephalus, or any of these guys, were they resonating chambers or were they heat exchangers? Well, we're pretty sure they were heat exchangers because there's big blood vessels that we can't explain otherwise. But I promise you, they also were resonating chambers because they couldn't avoid it. A large chamber will act as a resonating chamber, but consequently, animals will use that. We actually looked at the ontogeny, the growth of the crest in Lambeosaurs. We did that in collaboration with David Evans, who's a fabulous scientist at the Royal Ontario Museum. And it turns out that by looking at both the structure of their inner ear and their crest, the young ones were actually making higher pitched sounds, just like little kids tend to have higher voices. And so we're actually at, one of my former students is actually trying to actually see if we can calculate the resonant and vocal properties of crests in hadrosaurs, actually start to get frequency responses so that we can say things with more detail than just like low frequencies or high frequencies.
That is incredible. I'm going to hold my questions till another time because I've got lots.
What is your opinion on the biology and behavior of animals in prehistoric planet?
Yeah, fantastic, fantastic show. And I know the chief scientific advisor, Darren Naish, well. I've known him since he was a kid, or at least a grad student. We've co-authored together. We co-authored recently on some British spinosaurs earlier this year. And he is amazing, actually. He's a person that really digs into how animals work. What they did, they took some liberties. They looked at animals, and Darren was pretty good about trying to find at least some kind of examples that animals do the things they did in prehistoric planets. So he tried to do that. And, after the fact, he was, on Twitter and other things saying like, see, here's an animal that's doing this crazy thing that we had a dinosaur doing. So there are animals that do that. Yes, it might not be common, but it has been recorded that animals that are, reptiles or birds or something like that do something that's similar to that. I mean, in general, Prehistoric Planet obviously set new standards for animation, for realism. And I was grateful that Darren Naish was on board to make the biology as solid as it was. I mean, they went a little far in some cases, a little further than I might have gone.
Like the air sacs on Alamosaurus.
Yeah, that one was, and I think that they're sort of feeling like, I think we may have kind of gone a little too far with that one. That's about the only one that I would kind of say like, yeah, I'm pretty sure they couldn't do that. But there are, because they were actually using, and I can tell you why that one was unlikely, is that the air sacs that these animals have are actually relatively deep. There are birds, a lot of birds actually, that have what are called subcutaneous air sacs that are actually just below the skin. So that's what subcutaneous means. And so those are connected to the lungs. Some of them are actually connected to the nasal cavity. And so that they can actually inflate those, and those are done for displays, just like they were showing in Alamosaurus. And so they kind of took a little bit of liberty. But I'll go back to something that I said before, which there's maybe the grumpy me that we watched a couple of those things and kind of going like, I don't know, Darren, I'm not sure I'm bored with that one. But then I'd try to sort of say like, yeah, these were weird animals though. So some of these animals could have had things that we don't see today, right? We're looking at a narrow time slice right now. And dinosaurs were these crazy animals that we don't have, that are not like them. I mean, people like me tend to paint them in very much a modern brush. In other words, we look at the structures that they have, that their modern day relatives have, and we reconstruct them. But like I said, I'm always trying to do that, but still allow their own novelty to emerge. I don't want to squelch their uniqueness. So some of the stuff that they did were crazy. You know, my initial response was, I don't know, but then I'm kind of going like, maybe. Never say never with nature. I mean, if you just, I mean, the social media has been great because people will post these videos of animals doing wacko stuff that professionals like me kind of probably should have known they did, but didn't. And so the natural world continues to surprise us and delight us. And I would expect that the dinosaurs were doing the same. And I think on Prehistoric Planet, they were trying to deliver some of that, trying to deliver some of those wonderful things that that might have been exceptional even for dinosaurs.
What challenges do today's and future paleontologists face that you didn't?
There are scientific challenges that we're always going to have. Part of what I was referring to was the fact that I came up at a time where the fact that I was a white male from the northeastern part of the United States made things easier for me. And that was wrong then, it's wrong now, and our culture is working to correct that. And I think that we're making some good strides. So that's what I was sort of saying before, how about some doors were open for me that they weren't for other people. And we're doing better. But scientifically, we're having new advances all the time. One of the things that's sort of tedious and dry, but is actually a major revolution going on, is now in statistics. There's all of these statistical procedures that are going on right now that are allowing us to test hypotheses and really start to evaluate things in rigorous scientific ways that we couldn't before. And so, you know, in the past, you know, folks like me could just sort of stand up and say like, hey, I found a thing, let me show you. Nowadays, it's sort of like, okay, well, go back and find like, you know, 40 of those things and run a complicated analysis that's, you know, doing all of these other things. And, you know, that's makes it harder for us, but it strengthens the scientific inferences that come from that. So I can tell you that one of the things that's happened, statistics has always been around, of course. You know, we've been always like, you know, making, you know, drawing lines and, you know, averages and standard deviations and things like that. And we have other things like, you know, analyses of variance that have been around for generations. But, you know, coding in R, that might be a phrase that many of you have heard, coding in R. That has become a thing now that a lot of people spend a lot of time doing. They say lots of bad words at various times as they're struggling and their R code is not working and there's bugs in the code and all of that kind of stuff. And so a lot of science is actually now with coding and statistics. In fact, there are people that specialize with that. I work with people that specialize in that. I don't specialize in that myself. I'm often brought in as the anatomist on board, and we often have people that are, you know, geologists and people that are statistics people.
What new ways can engineering contribute to and diversify paleontology?
You mentioned engineering. Engineering has been a revolution for vertebrate paleontology science in general, where we have basically roped in all of these approaches. And it comes back to CT scanning, but also the virtualization of biology. In other words, we can take biological things and pull them into a computer environment. I mean, a lot of these statistical models are models that are run in computers. The anatomical stuff we do are models. I was showing you that Diplodocus with the muscles, you know, jaws were moving and the eyes were moving around. Those are anatomical models, but we are coupling those with engineering models. So are things that are looking at stresses and strains called finite element analysis. I mentioned I had a student, Jason Burke, who became an expert in modeling airflow through the nasal cavity, something called computational fluid dynamics, which is used in you know, doing in plumbing and airplanes and things like that. And he did pioneering work. He did the first analyses of nasal airflow in any reptiles, any bird, any extinct animal. You know, and he's he's not that old. So this is new stuff. And so the idea that we can actually start to test hypotheses using simulation has really started to emerge. There are some folks that kind of go like, yeah, well, that's testing a model, not really testing on a real animal. But the reality is, give me a break. We're doing the best we can. These guys are extinct. At some point you have to give us that. We're trying here. And what's exciting right now is that we don't really know what the limits of those are. And so we see lots of, people sometimes use the metaphor of a pendulum swinging. And so there was this time where the pendulum swung and people were doing all of these really cool studies. And the pendulum has swung back a little bit, sort of saying like, okay, well, wait a second. Here are these concerns that are there that we need to fix. We need to address some of these methodological or assumptions or some of these scientific things, and address those so that we can sort of hone down what we're actually saying. And then hopefully this pendulum will swing back and we can start to do even more things. And so there's always a push and pull in science, in anything really, a push and pull, where things start to move and then other people say, well, wait, not so fast. Here are these other things that are going on. And that probably happens in all segments of human endeavor. And science is no different.
Do troodontid eyes and ears indicate they could hunt in low light? What do their brains tell us about ecology?
Those are entirely the kind of questions that we're asking. I mean, that's we're in a sense working on that. Got a major project on dinosaur brains right now. Yeah, we're trying to look at all of that. So we can look at the inner ear, and that's going to be telling us stuff, and it tells us that we've got a really highly attuned hearing organ for sure, the cochlea, which is the hearing organ. But we can also see the eyes that are absolutely enormous. We can also look at the parts of the brain that are associated with processing visual information. So once again, we can look at the eye socket, the bony orbit, that tells us something. We can look at the optic nerve, the size of the bony optic nerve canal, which tells us something. But we can also look at the structure of the brain itself. And fortunately, troodontids are one of the groups of dinosaurs that actually fills most of their bony brain cavity with brains, such that the brain endocast is a fair reproduction of the surface of the brain in life, which is great. Many dinosaurs don't do that. Sauropods are among them, sadly. So all of this information is lining up to show us that troodons very much relied on the visual system as well as the hearing system. Their smelling's not bad either, really. And so these are small predatory animals, which we would predict, probably just looking at them, that they probably were pretty capable in terms of sensation and sensing their environment and processing that. But another thing that's super cool, and this gets back into gets out of my lab and gets into the other stuff that more conventional paleontologists do, which is actually where are these things found? Where do we find troodontids? Well, we find troodontids pretty much everywhere, but we also find them in interesting places. We find them north of the Arctic Circle, north of the Cretaceous Arctic Circle. These are animals that actually were living in periods of total darkness. And so maybe the Arctic wasn't quite as cold, although we pretty much think that there are parts of the Cretaceous Arctic that got snow. Maybe not, obviously nowhere near as much snow as they got now. But one thing they can't escape is the tilt of the axes, and it was dark. It was dark for months on end up there. And so as a result, we know that we would presume that they might have had some sort of adaptations for low light vision. And so we can look at their large eyes. We can look. Unfortunately, we don't have really good evidence of the bony eye ring, the scleral ring, in most of the troodontids, and certainly none of the Arctic troodontids. We don't have any Antarctic troodontids yet. But we do have them from the Arctic. And so it'd be interesting to see if they in fact had a bony eye ring that we associate with low light vision. And so it's it's the kind of thing that these animals may have been nocturnal animals in areas where they had normal seasons, but it allowed them, that low light vision allowed them to, in a sense, invade these unusual environments, these seasonally low light environments like the Arctic. And all of these things sort of fit. And it's one of the things, some of the next step things that we're trying to do is we're looking at some of the physiological things and how animals deal with heat. One area that we haven't looked super hard at right now is actually trying to make comparisons between, say, species that live in lush lowlands versus highly seasonal arid areas. Like the Morrison Formation is classically highly seasonal hot, and there's other environments that are much less arid and less pronounced seasonality. And so it'd be interesting to see, because there are going to be different environmental pressures on those animals, which should be tracked physiologically. And what we don't know yet, because we haven't done those studies yet, is whether we're seeing that in for example, looking at brain structure. Are we actually seeing that? Are we seeing enhanced brain cooling mechanisms in animals that are living in arid areas versus areas that are living in areas where there's lots more moisture?
How applicable is that for oviraptorosaur vision?
Raptorosaurs, again, I mean, almost all of these theropods were visually oriented, and almost all dinosaurs, as far as we can tell, were visually oriented animals. In part, we can just see that without even looking at their skulls or looking at their brains or anything like that. We just see, horns and spikes and frills and all of these things. It might have been for defense, but a lot of them are fairly clearly for display as well. They may have had a role in defense, but they also have a role in display. It's sort of funny to think that we humans do that too, right? We actually have weapons that are used as weapons, but we also have weapons that are actually used as display. We actually have these highly ornate swords and things like that. And there's the classic, we don't do it in this country, but some countries have these big military parades where they take their weapons and they actually parade them down the street. And so, in a sense, this notion of having weapons that you display is something that animals do too. And the idea is the same that we humans often have, is if we display our weapons in elaborate ways, we might not have to use them. And using weapons yourself is dangerous. It's dangerous for the other one, but anytime anybody's using weapons, it's dangerous. And so anything you can do to de-escalate these things, animals will do. And so they may have weapons that they can use, but they often are associated with display structures that, in a sense, make them use less, simply because they're sort of behavioral things that come in. So almost all dinosaurs were visually oriented. Oviraptorosaurs were visually oriented, and many of them, in fact, have these crests on top of their heads. Not all Oviraptorosaurs had crests, but many of them had bony crests. Some of them look like they might have actually had soft tissue crests. Things like Conchoraptor actually has a little sort of pneumatic open spot on top of their head that is, you just look at the skull and it looks flat, but I'm not sure, there might have been something going on up there. And we would expect that many of these dinosaurs would have had, wattles and combs and snoods and all of these things that we see in reptiles and birds today that are visual structures, as well as thermoregulatory structures. It's useful to keep those two in mind.
Do dinosaur brains and inner ears reveal much about behavior and ecology, including when they're most active?
Yeah, I sure hope so, because that's what we're spending a lot of time and money on. Yeah, I mean, at some level, behavior emerges from the brain, right? It's the brain that's the control system. It's it's the computer in the cockpit that's telling the body to do these other things. And so at some level, the brain's got to be important in determining behavior. It's also where all of this information gets processed. So all of this sensory information that comes in, whether it's visual or auditory or touch. We sometimes don't think about the importance of tactile information, but it's huge. And so all that information is coming into the brain. It's coordinating with, the motor system. And so the brain is mediating, managing all of that stuff. And so in a sense, the brain is there. The kicker for us is, number one, all we get is the surface of the brain. Even in something like Troodon, where we have a pretty brain-like brain endocast, it's just the surface. So we don't have, it's trying to do neurology without neurons. We don't have the things that are actually wiring these things together. And the reality is, even if you did that, even if you had the brain of T-Rex sitting right in front of you, it's hard to go from looking at the structure of a brain, even with all of its, neurons and myelin and, nerve tracts and all of that stuff, to go from behavior. You can take the brain of a tiger and the brain of a lion and put them right next to each other and then mix them up, and you wouldn't be able to distinguish them. And so it's not like you can sort of say like, oh, you know what? This one must be a lion because here's the social hunting lobe of the brain. And the tiger lacks that. It's a solitary hunter. It's lacking the social hunting, pride, pack hunting behavior. The reality is the brains look the same. And a lot of the behavioral flexibility that we see that animals have is done in the wiring. And so when I talk about painting a picture of the past, I always, I'm always reaching for, metaphorically anyways, a fine brush. And then I kind of go like, nope, drop the fine brush, dude. The best you can do is to paint with a broad brush. And so I can't tell you, how far away T-Rex could smell blood. I can tell you that T-Rex, the sense of smell was more important for T-Rex than it was for, say, Allosaurus, because it invested more neural tissue in the sense of smell. What was it doing with that sense of smell? I'm not sure. Maybe it was smelling prey. Maybe it was smelling the pee or poop of another tyrannosaur as it's carving out territory, or smelling the reproductive smells, trying to smell whether, females are ready to mate. And so there's all of these different things that we could potentially say. All I can say is that T-Rex had a sense of smell that was important to it. It was more important for T-Rex than it was for Coelophysis or for Allosaurus or for Troodon. And so what it was doing with those, I got some ideas, but I'm still short a time machine. And so, but we can start to rule some of these things out. So we would expect that T-Rex's capabilities olfactory capabilities were more important or they were better than for something like Allosaurus. And so, we can go along with the same thing with the visual structures. We can figure out what's more important for some of these animals. One of the things we're really interested in right now is actually looking at brain structures. I've got a grant with colleagues in Europe. Steve Bersati, who's one who I'm sure you've heard of. He's a paleontologist, of course, famous paleontologist. We're also working with cognitive zoologists that actually study animal cognition, run emus and ostriches through tests, and we're working with an actual neuroscientist who studies brains, and we're actually trying to study cognition, intelligence in some sense, problem solving, flexibility, learning, things like that. We're trying to do that in deep time. The project is called Deep Time Cognition. And so we're trying to look at stuff. So if we look at something like this duck-billed dinosaur right here, again, this is a juvenile Parasaurolophus right here. It's got a really large bulbous cerebral hemispheres. These are the higher centers of the brain, which tend to be where a lot of the cognitive functions are located: learning, behavioral flexibility, planning, things like that. And so some of these, we're starting to make some of these comparisons. We can compare it with something like the Diplodocus right here, which has a famously small brain. And a lot of what we're looking at right here is actually blood vessels that are actually sitting in there. And so the brain and the cerebral hemispheres were really quite small. Don't judge him. He was fine. But the cognitive abilities of something like the Diplodocus were almost certainly much more modest than something like Parasaurolophus which is an animal that has zero armor, right? It's got no protection, really. Its width may have been important versus something like Troodon here, which has very expanded cerebral hemispheres and probably was a very clever animal that had really good capacity for learning and planning and all of those things. Could Troodon use tools? I'm not saying no. And so, I mean, it's the rudimentary stuff, and tool use in animals is like a little different than, what we do. But, these are the questions that we're trying to ask. And once again, the question becomes, where is that wall of what we can know? And how can we push that wall back so that we can know more? And so we're getting there. And we're actually, this project gets back to, and one of the major parts of it is actually doing this giant statistical analysis where we can actually use large data sets and start to put some of these pieces together so we can get some scientific justification, rather than me going like, wow, that looks big, he must be smart. That's, something. And back in the, 30 years ago, that was as far as we could go. We can do better now, and we are doing better. That's part of what the science is doing.
Awesome. 2 questions left.
Have you studied cranial bite marks and pathologies? Do CT scans make them more apparent than bone alone?
Yes, the answer is yes. We've looked at pathologies. So we've got a project we're presenting at the Society of Vertebrate Paleontology meeting on another new pathology for Big Al. If you've heard of the Ballad of Big Al, the Allosaurus, we've got a new pathology in its mouth. So you're hearing it here first. We're going to be presenting it at the meeting, so everybody will know soon. So yeah, and the reality is, what kind of evidence we use, to be honest, as a paleontologist, it's always all of the above. I mean, paleontology is this detective thing where we're looking at any clues wherever they come from. Absolutely looking at the actual fossils. So yes, we do tons of CT scanning. We couldn't live without it. But we also go to museums and look at the specimens. And the actual fossils themselves are really important. But we also want to have CT scans of them or surface scans and all of those. So, I mean, we were looking at that with the T-Rex holotype with Emily. We've got some videos on that we did with Dissecting with Emily videos, looking at some of the pathology of the T-Rex holotype. T-Rex is becoming pretty famous for this face biting thing, which is just crazy, as if T-Rex wasn't terrifying enough. They just bite each other in the face. And probably more dinosaurs were doing that too. I mean, you can probably see dogs. I mean, you look at dogs playing and they're biting each other in the face. I mean, it makes sense, right? These animals, their heads are the point of aggression. So biting each other in the face is probably something that lots of animals did. T-Rex could do it and bite hard enough to cause damage. And so, we're interested in all of those. We're interested in, because you can tell things that are biologically relevant. Some of these things are sort of like, we can see evidence that, with pathology, sometimes the pathologies don't tell us that much that's grossly interesting, in the sense that, like, you can see lots of healed rib fractures in say, Sue, the T-Rex at the Field Museum. There's these calluses on the broken ribs. Well, I got tons of modern day specimens that come into my lab, birds and reptiles, and they got healed fractures all over the place. Animals get hurt, and basically how I sort of view it as they have some horrible injury that comes to them, and they kind of go like, I'm not dead, and I'm kind of hungry, so I guess I'm just going to have to figure this out. And the reality is they figure it out. I mean, a lot of these animals deal with massive wounds that they recover from. And so when I look at some of these dinosaurs and I go like, oh, wow, there's this healed wound, I kind of go like, well, okay, well, that's what animals do. They get injured because it's a rough and tumble life. And sometimes they die, but a lot of times they don't. And so they heal, and it probably sucks for them, and they limp around, and, they might not be as effective, in their daily lives, but they carry on.
What's your opinion on comparing dinosaurs to mammals?
Absolutely. Yeah, I do. I'll go back to what I just said. All of the above, man. I mean, take your comparisons where you get. The reality is that animals have to solve problems. And what's interesting about a lot of mammals is they get into the size range. So the skull of Diplodocus is about like this. which is about the size of a horse. Of course, the rest of the body of Diplodocus is much bigger. But I mean, a lot of mammals, studied rhinos, and all kinds of manatees. We've studied lots of large mammals, and they are in the size range of dinosaurs. And also we have the same parts. I mean, I mentioned like the maxillary division of the trigeminal nerve that, like T-Rex has. We have that too, which is why me as a dinosaur paleontologist can do just fine teaching human anatomy to medical students, as we all have the same parts. They're just arranged a little differently. And so mammals are absolutely highly relevant. We don't want to take any animal and just graft what they have onto dinosaurs. And so this method that I've been using over the years, this extant phylogenetic bracket, this EPB thing, where we actually look at modern day animals to try to frame and gauge the strength of our inferences. In other words, like how much of the features that they have are shared because of evolution, or did they evolve independently? So, for example, some attributes that we see in dinosaurs, modern birds and crocodiles and maybe even lizards have. And so we pretty much know that dinosaurs would have had those unless they lost them. And that's been very important for a lot of our studies of a lot of the fine anatomical details, like whether it's the jaw muscles or blood vessels or structures of the nasal cavity, a lot of these little tiny things that are important for us. We can actually see that a lot of these animals have them. It turns out that mammals might actually have something that's not what we would say is homologous. It's not actually shared due to common ancestry. Some of them are. Like I said, we have a lot of the same parts. But so, for example, the fact that mammals are warm-blooded, birds are warm-blooded. Those are separate evolutions of warm-bloodedness. And so it turns out that being an endothermic animal, a warm-blooded animal, has really important consequences for many aspects of your biology. And so a lot of the attributes that mammals have, birds have too. And so the question is, did dinosaurs have some of those things? And mammals become relevant. We always have to remember that that's an independent acquisition. That's an independent evolution. And so we can't necessarily graft mammal onto dinosaur, but we can recognize that mammals are responding to a lot of the same problems evolutionarily, and that they may independently come up with similar solutions. They may come up with different solutions too. And so that's part of our job as scientists, is to try to sort that out and actually revel in those differences, because they're often fascinating. I've got a student in my lab right now named DJ Morgan, who's really interested in marine reptiles, like mosasaurs and sauropterygians, plesiosaurs, things like that. And we're spending a lot of time looking at, we're looking at sea turtles and, water monitors and things like that, and crocodilians because they live in water. But we're also looking at cetaceans and pinnipeds, these aquatic mammals, because if you are swimming at depth, things like pressure, the hydrostatic pressure of the water is a big deal. You can't avoid that. And the cold, it gets cold really quickly. And so the pressure and temperature that affects you at depth is going to affect you whether you're a mammal or a reptile or whatever, or a penguin, a bird flying at depth. And so we can it's important for us. It's almost required that we look at these independent solutions to those unique problems, because a mosasaur would have had those same problems. For any of us that have in a pool dived to the deep end, we can feel that pressure on our ears. And it's like, dude, that's 12 feet. We're not talking like 400 meters or something like that, And so it doesn't take long before it gets cold and the pressures go up. And so people say like, oh, well, mosasaurs were swimming in the shallow seas of Kansas. And it's sort of like, yeah, it's cold and high pressure down there. It doesn't take very deep before you get that. And, you're basically a seal or a whale at that point in terms of experiencing that. And so they should have solutions to those problems. And that's one of the things that DJ has been looking at. So we're looking at all of these animals, all of the above. That's why it takes us a long time. You're saying like, hey, Whitmer, why haven't you published that damn paper on the scales on T-Rex? It's like, we're working on it, man. There's so many of these things. They're get in line. All of these things take time. And they're all fascinating. And we just chase all of these leads because they're all really interesting.
You live in a series of rabbit holes called a warren. All of your projects get lost. It's awesome.
Yeah, I mean, it's cool. I mean, we look at these modern day animals and they all have so dinosaurs or mosasaurs or whatever present us with these interesting questions. And we look at the modern day animals to help answer those questions. But the modern day animals have their own stories to tell that are fascinating. So, I mean, we looked at, for example, we looked at how rhinoceros horns attached to the skull, because it turns out that the bumpy bone that we see on rhinoceros skulls is pretty similar to what we see in things like abelosaurid theropods like Majungasaurus. We see giraffes have it too. And so I was trying to figure out what was going on with the skin of Majungasaurus, and we got giraffes in, and we got rhinos in, and we did a whole publication on rhino horn. It turns out, despite the name rhinoceros, which means nose horn, we didn't really know how the horn actually attached to the bone via the skin that's in between the two. And so, we did that, and it turned out to be really interesting. The word dinosaur doesn't appear. You don't need dinosaurs to make rhinoceroses. Really interesting. And so, rabbit hole, man, it's a warren. We're just going from one rabbit hole to another.
You got our last question, and I've got some wrap up.
Have your T-Rex scans given you a good sense of how intelligent a T-Rex may have been?
Yeah. So there's an article came out recently by Susanna Herculano Huzel, who's a fantastic scientist. Although this one article that she just published recently, if you've seen it got a lot of attention at the time, was actually saying that T-Rex was as smart as a baboon and probably had capacity for tool use. And a lot of scientists kind of went like, really? What's that based on? So a lot of people have been looking. I mean, I don't want to disparage, certainly don't want to disparage her because she's an outstanding scientist. You should check her out. She's a great public communicator. And so what we know about dinosaurs and how we analyze data about dinosaurs and dinosaur brains wasn't fully developed in her article. I'll just say that. And there's a number of people who have been looking at that. So once again, one of the challenges that we have is that all of the neurons are gone. And it's the neurons that are actually they are the chips, the microcomputers, the transistors, all that stuff that are actually creating the computer of a brain. We don't have any of that. So it's basically like, trying to judge how powerful a computer is by looking at the tower or looking at a laptop box and sort of saying like, what's in there? It's not quite that bad, but some days it feels like that. The reality is, and T-Rex is an interesting case because it has this brain endocast that has some attributes that looks like a brain. So there's the cerebral areas, these higher centers of the brain that I was pointing at before, like Introodon right here, are pretty big in T-Rex, but some of the other parts of the brain are harder for us to see. And part of that is because in T-Rex, and we're discovering in some other animals too, it turns out that modern day crocodilians is the same, is that the brain case grows longer than the brain itself, meaning that in a sense, the brain grows within the brain cavity, but then the brain stops growing or slows down faster, and the rest of the bony brain case grows bigger. So in a sense, the brain case, where we get these endocasts, in a sense, outgrows the brain. And so in a sense, as the animals get older, their brain endocast looks less and less like a brain. And so I can tell you that because we've actually looked at some juveniles. We did a study that was published in 2011 on a juvenile Tarbosaurus that was about this big. And we haven't published the brain yet, but we've got a brain of that. And I can tell you, it kind of looks like Troodon. In other words, it's a very bird-like brain, but as you get, and we've looked at older Tarbosaurus too, Tarbosaurus is basically the same as Tyrannosaurus, in case you're not familiar with Tarbosaurus. It's the same. Some people actually call Tarbosaurus Tyrannosaurus bataar. But the grown-up Tarbosaurus look like T-Rex, which are sort of brain-like, but not as much. But the juvenile is pretty brain-like. And so it gives us a better vision of, or a better view of what that brain was like. and it's fairly bird-like, but still fairly modest. And so the cognitive abilities of Triceratops, but Tyrannosaurus were probably better than what we would have thought years ago. They're probably much more impressive than we ever would have thought before, but probably not to the level of birds. Some people like to point to things like parrots and certainly crows. Parrots and crows are exceptional birds in terms of their cognitive abilities. I mean, these are amazing birds. These are birds that do things that most primates cannot do in terms of performing cognitive tests, tool use and multi-step learning and planning. They have capabilities that actually only some great apes can do at all. And in fact, that some humans can't do until they're like 3 or 4 years old. And so that's pretty impressive for a crow or a parrot. They can do math and things like that. Very impressive. Better than most other birds. And so just like higher primates tend to have greater cognitive abilities than, say, a deer or a cow or something like that, there's variation. So birds are, a crow or a parrot is smarter than an ostrich. But it looks like some of these small theropod dinosaurs might have been encroaching into sort of the rat-type range of cognitive abilities. Maybe some of them got super smart. It's one of these things that some of these dinosaurs, we don't really know that much about them. And so some of them could have been just like, if we didn't have a whole lot of, there's not all that many species of crows and parrots. And if we didn't know about them, we wouldn't know that they're these super smart birds. Feathered apes, sometimes people call those groups. And so some of these, there could be dinosaur groups that had really pretty substantial cognitive abilities. Tyrannosaurus probably was more modest. It probably was not baboon level, but it was way more than what we used to think. And part of this comes from, and maybe some of you are aware of this, one of the things that was discovered a number of years ago, and Susanna Herculano Husel was one of the people that discovered this, was that birds actually have a ton of small neurons. In other words, they have way more neurons than mammals do, and they're packed really, really tightly. And so, in fact, a bird that has a brain this big will have more neurons in it than a primate that has a brain that's like 3 or 4 or 5 times as large. And so, in a sense, that neuron packing, I've sort of likened it to that, birds in a sense have microchips in their skulls, and mammals sort of have these old-fashioned vacuum tubes. These older, we used to sort of think, how could a bird? And here's the brain of a golden eagle right here, which is a very impressive brain right here. But it's kind of small, right? We used to sort of think, how could birds? And people say that it's a bird brain. How could birds function with such small brains? How could dinosaurs function with such small brains? The new studies have actually sort of flipped that around and have now started to ask, why do mammals have such large, wasteful brains? Because it's apparently not, you don't necessarily need to have a brain that's big, and brains are very expensive. 20% of our energy actually goes to supplying our brain. And so there's tremendous costs to having a large brain. Why? So something like a sauropod having a small brain might actually be really smart, because a sauropod probably doesn't need to think about a whole lot of things. It's so big, it doesn't have to think a whole lot about predator defense. It eats plants that don't move. It basically has to recognize a member of its own species so it can mate, and maybe have enough cognitive ability to find nesting grounds and things. I'm sure they were even smarter than what we think. But the reality is, having a small brain energetically makes sense for a lot of animals. Being super smart and being able to think about stuff. We're humans, so we think like, shouldn't all animals be like us? But the reality is, why? Most animals are as smart as they need to be, and it doesn't make a whole lot of sense to be smarter than you than you need to be. It's a good question whether human, not to get philosophical, but I mean, we're so smart, and what good is it doing us? We're actually trashing the planet that we live on, making it almost impossible for us to think about thriving for all that much longer. I'm hoping we're going to figure it out, but this experiment in humanity, we don't really know what the results of that experiment is yet, and so it's interesting to sort of say, is our cognitive ability really all that desirable? But
build CT scanners.
Right. Yeah, exactly.
We have that going for us. Well, we are at the top of the next hour, so we're way over time. So Larry, thank you so much. The only other last questions in the chat, what T-Rex specimen is that over your shoulder?
Well, we got a bunch of them. So this one right here is, that's Bug, which is a little known common name for the most famous it was the most famous T-Rex for a long time. So this is AMNH 5027. So this is the specimen at the American Museum of Natural History that's been there for generations. It's been there for a century, seen by millions and millions of people. This is the model for the T-Rex in Jurassic Park. There's a Dissecting with Emily video that we did about that says why we know this is the model for Jurassic Park. And we've got a variety of other ones over there. We've got Gorgosaurus specimens, and we've got there's the Cleveland skull, whether that's Nanotyrannus or T-Rex. We've got Jane. We've got all kinds. We've got over 100 skulls of dinosaurs crammed into this lab right here.
Thank you, Larry, again.
My pleasure.
Back at some point.
Dr. Brian Curtice is a dinosaur paleontologist best known for his research on sauropods, the long-necked giants of the dinosaur world.
This animal is near and dear to my heart. This will probably be the name-bearing specimen for our new species of Diplodocus. And I dug this out. I'm not even humble bragging, I'm bragging. This took a long time to get out of the ground.
He has collaborated with scores of the world's leading paleontologists and has worked in hundreds of natural history, science, and dinosaur museums around the world.